Hello everyone, welcome to all. And today's topic is ABO blood grouping, Landsteiner rule, RH blood group system, and the importance of this blood group. I will discuss all the topics in details. If you like the video, please like, comment, and subscribe my channel. In the contents, we will discuss introduction about blood group, definition of blood, antigen and antibody, history of the blood group, Landsteiner rule, major and minor blood groups, ABO blood group system, principal and ABO agglutinogen, type of the blood group, concept of universal donor and universal recipient, RH blood group system, percentage of RH blood group in different blood groups, importance of the blood grouping and the RH typing. First of all, introduction. Blood group is also known as blood type. Blood group can be classified based on the presence or absence of the antigen on the surface of the RBCs and antibody present in the plasma. If specific antigen is present on the RBC, according to this, there is a specific name of this blood group. Then ABO blood group system is an important blood group system present in the human body, and it also presents some animals like chimpanzees, gorillas. Then there is a definition of blood. Blood is a fluid connective tissue in our circulatory system that plays various important functions in our body. Then antigen. Antigen are the protein molecules found in the surface of the red blood cell. In some blood groups, it may be carbohydrate, glycoprotein or glycolipid. Antigen is also called agglutinogen. Then antibody. Antibody are the protein present in our blood plasma that recognize foreign substance and active our immune system. Antibody is also known as immunoglobulin. So antigen is called agglutinogen and antibody are called immunoglobulin. History of blood group. Carl Landsteiner was first discovered the ABO blood group system in 1901, and for this he was awarded Nobel Prize in 1930. Adriano Strulli and Alfred von de Castello were first discovered ABO blood type in 1902. In the ABO blood group, there is a four type of blood A, B, ABO, and they are discovered these AB blood types in 1902. And these four blood types, Jansky was first classified the ABO blood group into four types, means A, B, ABO in 1907. And last of all, Alexander S. Wiener discovered Rh factor in 1930. Landsteiner rule. Scientist Landsteiner, after discovered the ABO blood group, he proposed two rules about these blood groups. First one, if an agglutinogen is present on the red blood cell membrane of an individual, the corresponding agglutinin or antibody will be absent in the plasma. Second one is, if agglutinogen is absent on the red blood cell membrane of an individual, the corresponding agglutinin will be absent in the plasma. So, if an agglutinogen or antigen is present on the red blood cell, its corresponding antibody will be absent. Or if the antigen is absent, so the corresponding antibody present in the plasma. In the next slide, when we discuss about the ABO blood group system, we will discuss about this rule more clearly. Scientists discovered more than 50 blood groups depends on the antigen and antibody and some of their characteristics. There are the some major and minor blood groups. So some name of the major blood groups are ABO blood groups and RH blood groups that we will discuss in next slide. And there is some minor blood groups present also in the blood. Lewis blood group system, Duffy blood group system, Kidd's blood group system, Kale blood group system, Bombay blood group system. ABO blood group system. Introduction. The ABO blood group system is more important blood types in the human blood transfusion. The ABO locus is located in chromosome number 9. It can be classified into four types based on the inherited properties of the red blood cell. One is blood group A, blood group B, blood group AB, and blood group O. Then what is the principle of this blood group? The principle of blood group is based on the agglutination. Agglutination occurs if an antigen is bound with its corresponding antibody, which is called isoagglutinin, and this process is called the agglutination. For example, when A antigen is bound with the antibody A, or when B antigen is bound with the antibody B, is called the agglutination. You can see in this picture there is a red blood cell and antibody in the plasma. When they are bound with each other, they form agglutination. ABO agglutinogen. In this picture, it is the structure of the RBC precursor, and there is the RBC is attached with the one glucose, two galactose, and one in acetyl glucosamine. But in case of H antigen, there is an extra molecule fructose that is attached with the galactose. So, the fructose containing H antigen is a basic antigen found in all individuals. So, H antigen we can found all the ABO blood group system, means A, B, AB and O. All types of the blood there is present H antigen. In antigen A, N-acetyl galactosamine as the terminal sugar on H antigen. In antigen B, the terminal sugar is galactose. And in ABO persons, both transferases are present, while in O blood group, none of the enzyme present. There is a three picture formation of H antigen, A antigen and B antigen. In the H antigen, you can see there is a fructose molecules that is attached with the precursor structure of RBC. And as the same way, in the A antigen, there is present N-acetyl galactosamine and fructose with the RBC precursor structure. And as the same way, there is a B antigen where there is a galactose and fructose that is attached with the precursor structure of RBC. So in case of A antigen, there is a N-acetyl galactosamine that is attached with the N antigen structure, and in the B antigen there is a galactose molecules that is attached with the H antigen and made their structure. I think this concept is clear to you all. Coming to the next slide, there is a types of blood group. First, blood group A. Blood having antigen A on the RBC cell surface and B antibody in the plasma belong to the A blood groups. Blood group B, blood having antigen B on the RBC cell surface and alpha antibody or A antibody is present in the plasma is belong to the B blood group. Then blood group AB. Both antigen A and B are present but serum does not contain any antibody is called AB blood group. Blood group O. Both antigen A and B are absent but alpha and beta antibody or A and B antibody are present in the plasma is called O blood group. It is a chart of the ABO blood group system. I think it helps you to understand all the blood groups and their antibody and antigen. So first, blood group A. In the blood group A, there is anti-B or antibody B present in the plasma and antigen A is present in the red blood cell. In the blood group B, there is the anti-A is present in the plasma and antigen B present in the red blood cell. So you can see in this chart that there is a opposite antigen antibody are present in a blood group. Corresponding antibody is not present in a same blood groups. Like there is the blood group A where anti-D present and A antigen is present, and blood group B where anti-A present and B antigen is present. As the same way, in the blood group AB, there is a none of antigen is present in the plasma, but both of the antigen A and B is present in the red blood cell. And blood group O, both antibody A and B is present in the blood group O, but none of the antigen is present in the red blood cell. So in AB blood groups there is no antibody is present in the plasma, and blood group O there is no antigen is present in the RBC. This is some important concept about the ABO blood groups. First is the universal donor. In the O blood groups patient, They have no antigen, so agglutination does not occur with any other type of blood groups. So O blood groups can given to any group of blood patient, and the pupil with this blood group is called the universal donor. Then the concept of universal recipient. In AB blood groups patient, plasma of the blood has no antibody. This does not causes agglutination of the red blood cell from any other types of blood groups. So the pupil with AB blood groups can receive blood groups from any blood groups person. So the pupil with this blood group is called universal recipients. Second important blood group system is RH blood group system that was discovered by the Karl Landsteiner and Wiener in the 1940. RH factor is an antigen that is present on the RH blood group system. It was first discovered in the rhesus monkey, so its name is RH factor. Then RH blood group is consist of the 50 different types of the antigen. Among them, there is a 6 most common type of antigen, and those are C, capital D, capital E, and small c, small d, and small e. And in these blood groups, mainly if the person is having D antigen is called RH positive. If without D antigen are Rh negative. Among Indian population, there is 85% of the peoples are Rh positive and only 15% of the peoples are Rh negative. Percentage of the Rh positive peoples is more among the black peoples. In the next point, Rh blood group system is different from ABO blood group system because antigen D does not have corresponding natural antibody. So if Rh positive blood is transfused to Rh negative persons, anti-D is developed in that person. On the other hand, there is no risk if Rh positive person receives Rh negative blood groups, because in the Rh negative blood there is no Rh factor is present in the blood groups. In this chart, I try to combine Rh factor with ABO blood group system. With the green mark, it detects Rh antigen, red marks can detect A antigen, and with the blue marks, it detects B antigen. In the Rh positive A blood groups, there is present Rh antigen and A antigen. In the B blood groups, there is present B antigen and Rh factors. In the AB blood groups, there is present A antigen, B antigen, and also Rh antigen. But in ORH positive blood groups, there is only Rh antigen is present. As the same way, in the Rh negative blood groups, there is no Rh factor is present. So in Rh negative A blood groups, there is only A antigen present. In the negative B blood groups, there is only B antigen present, and in the negative AB blood groups, there is only A and B antigen is present, but not the Rh factor is present. But in Rh negative blood groups, there is no antigen are present. In this slide, the percentage of the positive and negative Rh blood groups in the different ABO blood groups. Among the peoples, there is Rh positive O blood groups is 35.4% and negative blood group is 13.1%. In the A blood groups, there is 30.3% of positive and 8.1% of negative. In the B blood groups, there is 8.1% of positive and 2% of negative peoples. And in AB blood groups, there is a 2% of positive peoples and negative peoples is 0.7%. So in the worldwide, there is O blood group is present most of the peoples, and AB blood groups is present in the less amount of peoples. Importance of blood grouping and Rh typing. The first one is it helps blood transfusion. In paternity dispute, means it helps to identify the father of the baby. It also helps to prevent hemolytic disease of newborn baby and many medical legal cases in the susceptibility disease. Among the O blood groups, there is a most of the chances of the duodenal cancer, peptic ulcer, and in the blood groups A, there is a most of the chances in the gastric ulcer, carcinoma of stomach, pancreas, and the salivary glands. So thank you everyone. If today's session is helpful to you, please like, comment and subscribe my channel. That helps to grow my channel. And Must comment if there is any doubt. Thank you.